Welcome to our lecture on metabolomics. Today, we are going to explore the fascinating world of small molecules inside our bodies. By the end of this session, you will understand how scientists capture a real-time snapshot of cellular activity and use it to detect diseases long before physical symptoms appear. Let us dive in. To understand where metabolomics fits into biology, imagine a massive, bustling city. If you look at the city's original blueprints, you are looking at genomics, which shows what could potentially be built. The active work orders are transcriptomics, and the workers themselves represent proteomics. But if you want to know exactly what is happening right this very second, like the traffic jams, the pollution, or the goods being traded, you must look at the actual products moving through the streets. In biology, this real-time snapshot of cellular traffic is called metabolomics. Now that we have our city analogy, let us define this field formally. Metabolomics is the comprehensive, large-scale study of small molecules, which are known as metabolites, within a biological sample, such as a cell, tissue, or biofluid. These metabolites are the final downstream products of all the complex genetic and protein regulatory networks operating inside an organism. Common examples of these molecules include sugars, amino acids, lipids, and energy intermediates like ATP. Measuring them gives us the ultimate indicator of what is actually occurring in a living system. Building on those examples, it is crucial to understand that the metabolome is highly dynamic. Unlike your DNA, which remains mostly static throughout your life, your metabolite levels are constantly changing. They respond rapidly to physiological stimuli, environmental changes, or the onset of disease states. By analyzing this complete set of shifting small molecules, scientists can construct a highly accurate biochemical profile, or phenotype, of the organism. This means we can see exactly how an organism is functioning at one specific point in time. Understanding this dynamic profile is absolutely critical because it bridges the gap between our genetic makeup and our actual physical health. In the real world, metabolomics is heavily used in medical diagnostics to discover biomarkers. These are specific molecules that indicate the presence of a disease long before any physical symptoms appear. For instance, elevated levels of certain sugar metabolites in the blood can serve as an early warning sign for diabetes, allowing doctors to intervene much sooner than if they relied on genetic risks alone. So, how do we actually measure and identify these tiny molecules? Computational biologists and chemists frequently use a powerful technique called time-of-flight mass spectrometry. In this machine, metabolites from a biological sample are given an electrical charge and then accelerated through an electric field down a long vacuum tube. The crucial principle here is that the time it takes for a metabolite to reach the detector at the end of the tube depends entirely on its mass and its electrical charge. Let us look at the math behind this. This brings us to the fundamental mathematical principle behind the machine. We can calculate the exact travel time using the time-of-flight equation. In this formula, T represents the time of flight, measured in seconds. L stands for the length of the flight tube in meters. The variable m represents the mass of the metabolite ion in kilograms. Z is its electrical charge in coulombs, and V is the accelerating voltage applied by the machine in volts. Conceptually, this formula tells us that heavier metabolites, with a larger mass, will take a longer time to reach the detector, while a stronger voltage or higher charge will cause the metabolite to travel faster. Before we move on, let us see this with a concrete example. Imagine we are analyzing a blood sample to detect glucose. We are given a tube length of 1.5 meters, an accelerating voltage of 20,000 volts, a glucose mass of 3.0 times 10 to the negative 25 kilograms, and a standard positive charge. We substitute these known values into our equation. First, we calculate the denominator by multiplying the charge and voltage, giving us the kinetic energy. Dividing the mass by this energy gives us the value inside the square root. Taking the square root and multiplying by the tube length gives our final answer. Physically, this means the glucose molecule hits the detector in about 10 microseconds. Now that we have used this formula numerically, let us understand a deeper relationship it reveals. Suppose a researcher is comparing a heavy lipid molecule to a lighter lactic acid molecule. We are given that the heavy lipid has exactly 4 times the mass of the lactic acid. Both molecules experience the exact same charge, voltage, and tube length. Instead of plugging in messy scientific numbers, we set up the time of flight equation for the heavy lipid, substituting its mass with 4 times the mass of the lactic acid. This sets the stage for an algebraic comparison to see exactly how much longer the heavy molecule takes. Continuing with our derivation, we want to see how that factor of 4 affects the overall flight time. Using the rules of algebra, we can separate the number 4 from the rest of the fraction inside the square root. When we pull it out, we must take the square root of 4. Notice that what remains inside the parentheses is the exact original formula for the time of flight of the lighter lactic acid molecule. This mathematical manipulation is powerful because it allows us to directly compare the 2 flight times without needing any actual numerical measurements from the machine. So we have proven how the mass scales. The square root of 4 evaluates exactly to 2. By substituting the lactic acid time back into our equation, we arrive at our final result. The time for the lipid is exactly 2 times the time for the lactic acid. Physically, this beautifully illustrates the nonlinear relationship in mass spectrometry. The mass must quadruple just to double the travel time. This is a crucial concept that computational biologists use when writing algorithms to identify thousands of metabolites simultaneously from a single sample. To visually reinforce what we just learned and proven, let us look at the schematic of the mass spectrometer. On the far left, the biological sample enters the ionization chamber, where metabolites receive their charge. They are then pushed through the accelerating voltage grid into the long flight tube. Notice how the smaller, lighter molecules are racing ahead of the larger, heavier ones, exactly as our formula predicted. Finally, they strike the detector on the right, which sends a signal to the computer to generate a mass spectrum graph, recording their exact arrival times to identify each molecule. To wrap up our lecture, let us review the most important ideas. First, metabolomics provides a high-resolution, real-time picture of the biochemical activity happening inside a living organism. Second, by measuring these small molecules, scientists can discover biomarkers to detect diseases early and personalize medical treatments. Finally, instruments like the time of flight mass spectrometer rely on fundamental mathematical principles, specifically that travel time scales with the square root of mass. Mastering these concepts is essential for translating raw physical data into meaningful biological insights. Thank you for watching.